Hello everyone, in this video we are going to talk about the bacterial cell wall structure. So first we should know what is a bacteria. Bacteria is a prokaryote. Usually cells are divided into two kinds. One is prokaryote and one other one is eukaryote. प्रोकैरियोट वो ऑर्गेनिजम्स होते हैं जिनके अंदर जो न्यूक्लियस होता है दैट इज नॉट सराउंडेड बाय अ न्यूक्लियर मेम्ब्रेन। जैसे कि हम कह सकते हैं, अगर हम एक स्ट्रक्चर की बात करें दैट हाउ अ सेल लुक, व्हाट वी कैन से दैट दिस इज अ सेल। आई हैव मेड अ स्ट्रक्चर ऑफ सेल। Yeah, this is a cell, and that cell has a well defined nucleus in it. This nucleus is surrounded by what is called as a cytoplasm, and cytoplasm has various cell organelles present in it which perform different structures, like it contains mitochondria, endoplasmic reticulum, and many more. So the basic difference between a prokaryote and a eukaryote is that there is no well-defined nucleus in case of my prokaryotes, but there is a well-defined nucleus in case of my eukaryotes. So bacteria is a prokaryote. If we talk about the classification of organism, there are five kind of classification. One is Monera, second one is Protista, third one is fungi, fourth one is plantae, and fifth one is animalia. So monotrons are the one which belongs to prokaryotes, and other four belongs to eukaryotes. And our bacteria fall in this monotrons category. So this is some basic things that you should know about where the bacteria lies. It is a prokaryote organism which falls under the category of monotrons. We as a human being have the outermost cell membrane. हम ह्यूमन बीइंग हैं, हम लोगों की आउटर सेल मेम्ब्रेन होती है. दिस सेल मेम्ब्रेन इज मेड अप ऑफ फॉस्फोलिपिड. व्हाट इज दिस फॉस्फोलिपिड मीन्स? Lipid is anything which is mainly a fat, and fat is made up of fatty acids and my glycerol. So when fatty acid attached to the glycerol, it will make fat. And when we have attached this fatty acid with the phosphate group, we will make a phospholipid. This is the basic composition of a cell wall, whether it's a human cell wall, plant cell wall, animal cell wall, or the cell wall. This is the basic composition of cell membrane, not the cell wall. It's a basic composition of cell membrane. So this is about cell membrane. Next comes that in our human cells, there is the outermost layer would be a cell membrane. There would be nothing outside this cell membrane. But in various other organisms like bacteria, fungi, plant, there is a cell wall outside a cell membrane which help in maintaining the integrity of the cell. So we should know what does this cell wall of bacteria composed of. Bacteria are divided into two categories, which are gram positive and gram negative. This will depend upon the straining technique. There was a gram straining technique. So according to that classification, they have divided organism into two categories, bacteria into two categories. One is gram positive and one is gram negative. So the basic difference in this gram positive and gram negative lies in their cell wall, which we should need to understand. Talking about the gram positive bacteria की जो cell wall होती है, that is made up of peptidoglycan, and there is nothing outside this peptidoglycan. Whereas this peptidoglycan is very thick in case of gram positive bacteria. But in case of my gram negative bacteria, this peptidoglycan is thin, and outside this peptidoglycan there is one another layer which is called as outer membrane. And this outer membrane is most importantly made up of lipopolysaccharide, and this lipopolysaccharide is also called as my endotoxin. This is also called as pyrogen. So anything when we will call about pyrogen testing, pyrogen means anything which increases the body temperature of our body is called as pyrogen. So the endotoxin of the gram negative cell wall is called as the pyrogen, or it is called as lipopolysaccharide, which is mostly dangerous in case of gram negative bacteria. So first thing, the common between two gram positive and gram negative is a peptidoglycan, but the difference is that it is thicker in case of gram positive, whereas it is thickened, it's very thin in case of gram negative. So we should understand how this peptidoglycan look. What is the chemical composition of this peptidoglycan? Peptidoglycan. If we will talk about the meaning, if we break this peptidoglycan, it is peptide plus glycan. पेप्टाइड का मीनिंग होता है जब बहुत सारे अमीनो एसिड्स एक दूसरे के साथ ज्वाइन हैं थ्रू द पेप्टाइड बॉन्ड, वी विल कॉल इट अ प्रोटीन. सो पेप्टाइड टर्म आया, देयर इज सम अमीनो एसिड्स प्रेजेंट इन दिस लेयर. Glycan is a term which is used for the sugar molecules, कि एक चेन है sugar molecules की. So we get to know that peptidoglycan is made up of amino acids plus sugar. This sugar can be any sugar. So if we will talk about one peptidoglycan, it is made up of the repeating unit of NAM and NAG. What does NAM mean? NAM means N-acetyl muramic acid. N-acetyl muramic acid. And another one is NAG, which is N-acetyl glucosamine. These are the two kinds of sugar molecules which are present in my peptidoglycan. NAM is connected with the NAG with the help of a bond. This bond is called as transglycolase. Glycolase. This glyco term comes from glycan. So this is the bond between NAM and NAG. In case of NAM is attached with 5 amino acids. One is L-alanine, G-glutamine, L-lysine, G-alanine and D-alanine. 2 D-alanine, 1 L-lysine, 1 D-glutamine and fifth one is L-alanine. This is basically our structure of what is called as the gram नेगेटिव. ग्राम नेगेटिव जब हमारे बैक्टीरियाज होते हैं उनके अंदर ये 5 हमारे अमीनो एसिड्स प्रेजेंट होते हैं. लेकिन अगर हम यही बोले ग्राम पॉजिटिव की जो पेप्टिडोग्लाइकान है उनके अंदर एल लाइसिन नहीं होता, उसकी बजाय होता है डाई अमीनो pimalic acid. This is the component, this is the amino acid which is present in gram positive. This is the first difference between the two peptidoglycan. Next thing is that this is one peptidoglycan monomer. Many such monomer connect together to form a polymer. अभी क्या होता है, ये मोनोमर दूसरे मोनोमर से कनेक्ट होता है. पहला तो इस तरीके से ट्रांसग्लाइकोलेज बॉन्ड जिसकी हमने बात करी है. नेक्स्ट क्या होता है, ये डी एलेनिन बॉन्ड का ब्रेकडाउन होगा और जब भी बॉन्ड ब्रेक होता है देयर वुड बी अ रिलीज ऑफ एनर्जी. ये जो एनर्जी रिलीज होती है वो क्या करता है, वो अमीनो एसिड्स को ज्वाइन करता है, दो मोनोमर्स की अमीनो एसिड को ज्वाइन करता है जिस बॉन्ड को हम बोलते हैं ट्रांसपेप्टिडेज. So this is about the structure of peptidoglycan. These are the basic things that you should know. Next come the same thing is that mesodiaminopimalic acid as I have already discussed with you that this is the amino acid which is present in my gram positive, whereas in case of gram negative, which amino acid is present? It is L-lysine, and this is the transglycolase bond, and this bond, जो ये बॉन्ड बनता है दो अमीनो एसिड्स के बीच में, इसको हम बोलते हैं ट्रांसपेप्टिडेज। तो जब एक पेप्टिडोग्लाइकान का मोनोमर पेप्टिडोग्लाइकान के पॉलीमर में कन्वर्ट होता है, तो जो हमने बोला था पांच अमीनो एसिड से वो चेंज हो गए 4 अमीनो एसिड में। जो लास्ट वाला डी एलेनिन था वो रिमूव हो गया, और जो उसने एनर्जी रिलीज करी थी वो यूज हो गई ट्रांसपेप्टिडेज बनाने में। This is about all the basic things related to the peptidoglycan. Next come the point that there is one acid, teichoic acid, which is present in just gram-negative bacteria. This is not present in any of the gram-negative bacteria. This is present in gram-positive bacteria. This is the other difference between gram-positive and gram-negative bacteria. And the last difference is that there is the presence of outer membrane in my gram-negative bacteria, but there is no such membrane in case of my gram-positive bacteria. और जो ये ग्राम नेगेटिव में जो आउटर मेम्ब्रेन होता है उसका मोस्ट इम्पॉर्टेंट कंपोनेंट होता है लिपोपॉलीसेकेराइड, जिसका दूसरा नाम है एंडोटॉक्सिन। ठीक है, और इसी को हम पायरोजेन बोलते हैं, एंड दिस इज द थिंग विच कॉज डेंजर इन आवर बॉडी व्हेन एनी ग्राम नेगेटिव बैक्टीरिया कम्स इन आवर बॉडी। एक और टर्म होती है एक्सोटॉक्सिन। एंडो मतलब होता है जो अंदर प्रेजेंट होता है, बिकॉज ये सेल मेम्ब्रेन का कॉम्पोजिशन है तो हमने इसको एंडोटॉक्सिन बोला। लेकिन जब कोई ग्राम पॉजिटिव बैक्टीरिया हमारी बॉडी में आता है और वो क्या करता है, वो कुछ कंपाउंड्स रिलीज करता है विच आर कॉल्ड एज एक्सोटॉक्सिन। Those exotoxins are not the part of their cell wall. They are released by gram-positive bacteria. So exotoxin is released by gram-positive bacteria, and endotoxin is a part of outer membrane of the gram-negative bacteria. This is all about the things that we should know about the difference between my cell wall of gram positive and gram negative bacteria. With this, I will end up my video for today. Thank you so much for listening.